Hey guys, this video is going to go over how we make phylogenetic trees and cladograms and just the concepts of taxonomy, classification, and phylogenies. Don't forget at any time you can pause me or rewind if I'm going too quickly. I tried to color code the most important things, the things you really needed to write down, so look out for those in red. I might have missed a slide or two there. And if I do, then I'm just going to go and underline it in red. So here we go. So taxonomy and classification is what I like to refer to as naming and sorting. Taxonomy is all about naming the organism, so that's how we come up with scientific names. And classification is all about putting them into groups based on some structural similarities. Those similarities can be both inward and outward, so internal and external. The system itself was made up or came up with by a guy named Carolus Linnaeus, also known as Carol Linnaeus, and that happened in about the 18th century, so this has been around for a really long time. It is a hierarchical system, so the largest and most inclusive grouping, so the one that contains the most organisms, would be the kingdom. Okay, and if you look at the little diagram, then this is where we're starting. Okay, now as you go, in this case, up, we're getting more and more specific, so it's more and more smaller groupings. So then the most specific grouping would be the species. Okay, so hopefully you got that. Okay, so a species scientific name is Latin, and it's comprised of two names, the genus name followed by the species name, and it's written in a very specific way. And now either you write the entire thing in italics. If you can't write it in italics, then write it as normal and underline it. Only the genus name is capitalized. The species name is lowercase. Okay, so this would be an example of a scientific name. Axionex jubatus. That would be the scientific name for a cheetah. So taxonomy is all based on classification, which is based on shared characteristics from one organism to another. So within the last 10 years or so, the classification world has kind of been turned on its head because of another scientist named Carl Worst, who proposed, instead of getting or having our kingdoms be our most inclusive category, he went ahead and added the concept of a domain. So he proposed that there are 3 domains, and he based those domains on RNA differences between prokaryotes and eukaryotes. Well, eukaryotes had enough similarities that they're all one big domain of their own called eukarya. Oops, sorry. Whereas prokaryotes had enough differences that they could actually be split into 2 groups called archaea, which are fairly closely related to eukarya, and bacteria. Excuse me. So organisms are grouped from species to domain, the groupings again increasing the groupings becoming more inclusive, as you go up. So now the kingdom isn't the most inclusive group, it is now the domain that is the most inclusive group. And when you were in freshman biology, you might have learnt like an acronym, Dear King Philip came over for grape soda or good soup or whatever, grape sandwiches, I don't know. Whatever it is they taught you to help you remember the order in which these classification groups go. Again, domain is the biggest, and species is the smallest. Or you could think of it as most specific and It's the other way around. You can think of it as up here being least specific. So lots of inclusion. And down here being most specific. Okay, phylogenetic trees. So phylogeny is the study of evolutionary relationships among a group of organisms. Okay, what is the common link? And based on these studies, we can build phylogenetic trees. So they're pretty much just illustrations or structures that show the evolutionary relationships of a group of organisms. They're based on three things: morphology, which is structure, and also the fossil record. So we base them on morphology and the fossil record. We also base them on embryology and DNA, RNA, and protein similarities. Excuse me. So the two terms that you will see being thrown around a lot would be phylogenetic tree and cladogram. Well, what's the difference between the two? A cladogram is used to represent a hypothesis about the evolutionary history of a group of organisms. So I think the panda bear evolved, and then the red panda was next, and after that came the polar bear. That's my hypothesis. So I would use a cladogram to showcase that. A phylogenetic tree, on the other hand, represents a true evolutionary history of an organism, one that's been proven time and time again. So needless to say that there are a lot of cladograms and not as many phylogenetic trees. And to be honest, the more we delve into especially molecular biology, the more that these phylogenetic trees are changing. Okay? Right, so here's an example of one. This shows the evolutionary history of what we know as the elephant. And these are all of the organisms that are part of the elephant's evolutionary tree. So I want to point a couple of things out to you. First of all, a phylogenetic tree shows at the top, most recent, and at the bottom, it shows history, pretty much. Okay, so the furthest away. We can say ancient times. Okay. Wherever a branch stops, wherever it doesn't go all the way to the most recent timeline, like it does here and here and here and here, and a couple of other places, this organism is now extinct. Okay, excuse me. So any organism that's not actually up at the top is extinct. So in terms of what's alive, and is still part of this elephant's family tree, we're looking at hyraxes, which you can see at the zoo, manatees, and then we have Asian elephants, and then the two species of African elephants. Everything else is now extinct. Okay, so we can take cladograms and phylogenetic trees, both of them, and break them up into sections. So right here, where we go this way versus that way, that is called a node, or a branch point. And a node always gives rise to two lineages. Technically, it should only give rise to two lineages. If it ever gives rise to more than two lineages, like in the case right here, where we have polytomy, this is a hypothesis in work. You can think of it like that. This is an unresolved pattern of divergence. We haven't completely figured out who really goes where. So you could think of this as being true, and you can think of this as being hypothesis. Okay. So taxas or taxon, it's singular, taxa, plural, are just groups, just groupings of organisms that share what we think is a common ancestor. So like right here, we would call these sister taxa because their common ancestor would be here. Then they'd share another one there and another one here. Okay? Be aware that these nodes and these branching points can rotate, so we could literally have spin this completely around and put C here, and B will still be there, and A would be here, and it would still technically be the same thing. Alright, so this is just to show you what I meant about those branching points being able to be flipped around. This is just showing the same evolutionary relationship. We've just kind of rotated where certain taxas go. So like this guy was once down here, and they've exchanged places. So they can be flipped around and still show the same thing. Okay, so what's a clade? A clade is a taxon, so a group, that consists of all the evolutionary descendants of a common ancestor. So all of these things highlighted in those different colors, those would be clades. Once they share a common ancestor, they're part of a clade. Again, the common ancestor would be wherever we have that split. So there, and there, and there, and there. So now we're going to start talking some terminology. A true clade is what we call a monophyletic group, and it contains a common ancestor and all of the descendants of that common ancestor. So like right here, this is where the common ancestor would have been, and these are all of the organisms that are as a result of that common ancestor. Another term that I want you to learn is paraphyletic group, and it's one that has a common ancestor but does not contain all of the descendants. If you look at group 2, again, this would be the common ancestor, and these would be the offspring as a result, but here we've missed one. So that's why it would be a paraphyletic group. We're just missing one that really should be a part of that group. Then a polyphyletic group does not have a unique common ancestor for all of its descendants. Okay, so like all of these guys have a common ancestor right here. So this would be the common ancestor. But this guy is included in that grouping, and his common ancestor, at least with these, isn't until down here. That's where they would have a common, but we're looking at the most recent. Okay, we've already talked about anagenesis versus cladogenesis, but let's go over it one more time. So another way to think of anagenesis is as a phyletic change, and it's an accumulation of changes in one species that leads to speciation over time. It is an evolution of the entire population. So we started off with this bird, and over time, this guy died out or became extinct, and this bird is the replacement. Okay, so over time, we've changed some of our features, and we've become a completely new species, but we don't have two species anymore. We only have one species. This species is alive, and this one is dead. Okay? Again, cladogenesis results in way more biological diversity because you have the common ancestor still existing. So we started off with a common ancestor. He survives and is still a part of that ecosystem or that population or that community. But through time, he has budded off. Some of his characteristics have changed and have resulted in another species. But the common ancestor is still alive. So we have this guy being alive and that guy being alive. Normally what happens with cladogenesis is we have some kind of physical separation. So the group, so again, allopatric speciation. Sorry. Okay, so something physically separated them like a huge mountain or something, and prevented them from continuing to mate as one species. So, how do we recreate phylogenies? We start with our unknown. We start with the fossil record. And pretty much that's the only way we'll get information on ancestral species unless they are still around today. And that is not going to be every single species. So, note, the deeper down in the soil layer the fossils are found, the older that fossil is. And then we can start building on there in terms of, okay, how long ago was this guy around before this guy came around, and this guy, and this guy, and this guy. Excuse me. So, we're going to start using some terms that you should be familiar with. Once we have our fossils, and we can compare our fossils to our descendant species, we start comparing features. So we're going to look at features that the descendant species has and the common ancestor has. And those species are going to be homologous. Homologous features again can be heritable traits. So it might be anatomical structure of some sort. It could be DNA sequences or protein sequences or something along those lines. So during the course of evolution, you're going to have a changing in traits. It doesn't mean that the trait completely disappears, but it means that it gets altered. So the original trait is what we call the ancestral trait, and then the trait that has been evolved is what we call the derived trait. Again, any feature shared by two or more species that's inherited from a common ancestor is called a homologous trait. Okay, that leads us to the term analogous structures. So analogous structures are going to be those that are similar in terms of structure, what they look like, or maybe even function, but are not necessarily inherited from a common ancestor. So like the wings of a bat versus the wings of a bird. Not a whole lot of commonality there in terms of ancestorship. Now, in establishing a phylogeny, we do use homologous structures. We never, ever, ever use analogous structures. We do not use them to establish a phylogeny. Molecular clocks. So the only way you can have a structure, an anatomical structure, is if you have a gene that codes for that particular structure. So just like how we can see conservation of structures from ancestors to descendants, we also see a conservation of those genes that give rise to those structures. And these are called molecular clocks. So homologous structures are coded for by genes, and those genes are going to have a common origin. That would be the ancestor that everyone came from. These genes may still mutate over time, and if you think about it, they kind of have to mutate so that we can have those changes, but they still retain a lot of that common and ancestral DNA sequences. So through genomic sequencing, we're able to compare and identify what these molecular homologies are, and again, they're called molecular clocks. The more closely related two organisms are, the more their DNA sequences are going to be similar to each other or resemble each other. Okay, so we have something called the molecular clock hypothesis, and it says that among closely related species, a given gene usually evolves at a reasonably constant rate. And pretty much what we do is we use these mutations to predict where divergence took place evolutionarily. Excuse me. Right, so you don't really have to write anything. One of the ways you might see this represented is in some kind of graphical form. So this shows amino acid differences over a period of time. And this specifically is talking about the amino acids in hemoglobin. And we're looking at a period of 500 million years. The slope of your line is going to represent the average amount of change taking place within this particular molecular clock. And it just shows you that different genes evolve at different rates. So it's not just one thing necessarily affecting the rate of evolution. Okay, again, you don't have to write anything for this slide, but this just kind of shows you that molecular clock thing that I was talking about. So if you look at it, this excuse me, here is our common ancestor with his DNA sequence. And then over a period of time, nothing really changes until we hit this 25 million year mark. And this is just an example. But I want you to note that in this species, we're going to do a substitution of a T for a G, kind of in the middle of the sequence. And then down here, we're going to do another substitution, but this time we're going to substitute a G for a T at the end of the sequence. So two mutations occurred. They occurred at the same time period, but it didn't result in the same change. Okay, so then we're going to keep going. At about 50 million years, we're going to notice another mutation is going to take place. So remember that G from before, that's still there, but the T in front of it has now been switched for a C, so another substitution. And down here at the same time period, another T, a T has been switched for a C. Again, two more mutations at the same point in time, two different results. So if this continues to happen, we're going to have two separate species, both originating from this common ancestor. But if you look at it, it took 50 million years, and we've had two base sequence changes, just two mutations. All right, we use molecular clocks a lot in the study of genomes and the study of especially viruses that really reproduce quickly and really mutate quickly. So like HIV would be an example of that. Based on these studies, we can determine that HIV kind of entered the human population in the 1960s, but the origin of the virus dates all the way back to 1930, so it's not 1980 like everyone seems to think. Another graphic on molecular clocks. You can use molecular clocks to produce phylogenetic trees. Okay, so how do we make a phylogeny? How do we go about constructing one of our own? Well, there are two rules that you have to follow. The first rule is called the rule of maximum likelihood, and the second one is called the rule of maximum parsimony. Well, maximum likelihood says that when you think of all of the different hypotheses that you have in front of you, take into account the hypothesis that reflects the most likely sequence of evolutionary events, while keeping in mind how DNA changes over time. So in other words, choose the one that makes the most sense. And rule number 2, parsimony, says that when considering multiple explanations for an observation, first investigate the simplest explanation that is consistent with the facts. So, start simple. Alright, so those are your two rules. Choose the one that makes the most sense, start simple. So you're going to see this represented in a whole bunch of different ways, where you'll have a table, and that table will show the differences between however many organisms you're looking at for whatever trait it is you're looking at. Your tree should reflect your table. So if you look at it, between humans and mushrooms, there's 30% difference, versus humans and tulips, where there's 40% difference. So the one that makes the most logical sense tells us that humans and mushrooms should be close to each other, okay? Whereas humans and tulips should be furthest away from each other, because they have the most differences in between, okay? If you also look at it, between mushrooms and tulips, we know that there's 40% difference, but it's still telling us that mushrooms are kind of in the middle. They're in the middle of the human and the tulip. So whatever tree you generate should be the one that showcases the mushroom in the middle. Okay, so how do you make a cladogram? You do it based on traits. Especially in class and even on the AP test, you're not going to have a whole bunch of fancy equipment to work with. You're working with pretty much what you can see. So you start off by examining the data given to you. Then you propose a cladogram that shows the evolutionary history that makes the most sense. All right, so here's an example using some vertebrate animals, and here are what we're comparing. We're looking at backbone, hinged jaw, walking legs, and do they have an amniotic shell or an egg, whatever, and hair. Again, these are just being compared one to the other. So we're comparing a lancelet, which is a very primitive type of fish, a lamprey, tuna, salamander, turtle, and leopard. Excuse me. So first you want to figure out which is your outgroup, which is the one that is not as closely related to everyone else. This is going to be the lancelet. Look at that. Not a whole lot of similarity here. Zeros in every column. Okay, so this is what we call our outgroup. And from there, it's kind of just a matter of common sense. So, does a lamprey have a backbone? Yes, it does. And you may not know what all of these animals are, but you just kind of have to go with, with the one that makes the most logical sense. So if you look at it, a 1 indicates that you have that trait. So everything has a backbone. When we get to hinged jaws, notice that our lamprey drops out. But from tuna all the way up through leopard, they all have hinged jaws. When we get to 4 walking legs, I mean, think about it, tuna's a fish, no walking legs there, so it drops out. Everything from a salamander up to a leopard stays. When we get to the amniotic shell, hopefully you knew that a salamander is an amphibian, which means that they lay their eggs in water, so their eggs aren't shelled at all, so the salamander drops out. But we still have our turtle, we still have our turtle and our leopard. When we get to hair, well, there goes our turtle because he's a reptile. He has no hair, and all we have left would be our leopard. So now we have to actually build the cladogram. So how do we go about doing that? The same way we analyze this table. You look at what you're given. So this is an example of what a cladogram of those same organisms would look like. Every branching point is almost like a stop sign for, vertebral column, no vertebral column. If you have a vertebral column, please go to the right. If you don't have a vertebral column, please go to the left. So you kind of have to think of it as almost like a policeman calling traffic. You have this, go this way. You don't have that, go that way, until you have your cladogram built. The one thing I can tell you is you get good at cladograms by building them and by practicing them, not through notes. I had to give you basic information. We're going to spend the next couple of days just practicing. So hope this was helpful. See you guys soon. Bye-bye.